Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled, The Essential Elements of Fast, Accurate, Quantitative Amino Acid Analysis of Biopharmaceuticals. Amino acid analysis is a critical and necessary function in quality control and process monitoring operations. It also has applications in protein and peptide identification and in the quantitation of a biotherapeutic API and finished drug product. Today's webinar has been designed to demonstrate how the use of two innovative technologies, ACQUITY UPLC and AccuTag Ultra chemistries, can greatly enhance researchers' abilities to accurately conduct the quantitative amino acid analysis of biopharmaceuticals. I'm John Sturrow, Editor-in-Chief of GEN, and I will serve as the moderator. After our speaker, whom I'll introduce in a moment, completes his presentation, there will be a question and answer segment. Feel free to send in a question for our panelist at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. Our speaker will try to answer as many questions as possible. Now, why don't we meet him? Dr. Tom Wheat is Principal Scientist in the Systems Laboratory at Waters Corporation. Tom has been involved with the development of several novel technologies for purification and isolation, as well as tools for protein analysis. A prolific author, he has published and or presented over 100 papers in the fields of analytical biochemistry, capillary electrophoresis, mass spectrometry, and preparative chromatography. I know Tom is ready to begin his presentation, so I'm going to turn the program over to him. Tom?
Thank you, John. Today I'd like to discuss with the group some of the work that we've been doing with amino acid analysis really over the last few years, and specifically the things that we've been doing that are important in biopharmaceutical analysis. Our work is centered upon a separation of the amino acids using UPLC of the amino acids and giving us a reliable analytical method. You'll notice in our basic chromatogram that all of the peaks are well separated, well identified, and sit on a flat baseline, which makes the quantitation very easy. To consider how we use this and what this means for us, I want to go back a little bit into the basics of amino acid analysis. Amino acid analysis is used in a lot of different ways, and it's required in a lot of different disciplines and applications. We do want to draw a distinction between amino acids as components of proteins, where we often refer to the amino acids as bound intermediates, but also as metabolic intermediates, which are nutrients that are used in the synthesis of proteins and are also used in general pathways for particularly moving nitrogen and other functional groups around. It's a particularly difficult analytical problem because the separation requires that we deal in a single method with a very wide range of chemical properties that includes very small differences between pairs of analytes. At the same time, the molecules do not have a common detectable element and no chromophore and occur over a wide concentration range. This problem has been addressed in a number of ways over the years. The separation of amino acids usually occurs in one of two ways, either cation exchange chromatography or reverse phase HPLC. Cation exchange is the classical original method for doing complete amino acid analysis. It exists today and is still used today because of the large historical database. It is a method that typically takes a half an hour or an hour to run a sample. In the last few decades, it has become very common to use reverse phase HPLC of derivatized amino acids. This technique for doing the separation gives us the best selectivity in the separation because we're using all the properties of the side chains. Because of the derivatization, we also can get the best sensitivity. And this combination of an easy separation leads us pretty directly to the shortest run times. Once the amino acids are separated, however, we still have to monitor the separation for purposes of detection and quantitation. And there are two basic approaches. One, of course, is the post-column reaction, used always in conjunction with ion exchange, but possible with other separation techniques, where a reagent is added to the effluent stream from the column in order to make the amino acids have a common detection. This we often think of as an anhydrin device, but we've also seen some application of pulsed amperometric detection and electrospray mass spec. The pre-column derivatization is perhaps the most generally applicable and easiest to maintain. There are literally dozens of reagents that have been used for amino acid derivatization, and those many reagents all work. Many of them have been commercialized, and they all improve the chromatography at the same time that they add detectability. This leads to the faster and more sensitive separation, but using these things does require some attention to the details of the derivatization chemistry. When we embarked on our work, we wanted to have some fairly specific goals in mind. We wanted to use a well-established reagent, our AccuTag reagent, because we understood the reaction and how to control it. We wanted to use our ACQUITY UPLC system and H-class systems because we could get the highest resolution by virtue of using ultra-performance liquid chromatography. We also wanted to control the composition of the eluents so that the method could be effectively a turnkey method. Finally, we put the new method based on our Empower software because it is compliance ready, but also because that made it possible for us to provide method templates and to provide custom calculations. The method that we develop is based on the AccuTag derivatization chemistry. The AQC reagent reacts with primary and secondary amino acids to give the same derivative with very nearly the same extinction coefficient. The excess reagent reacts with water very quickly to give aminoquinoline and then hydroxysuccinimide, byproducts that do not interfere with the separation. There is a reaction of the reagent with AMQ to give what we label in the chromatogram as a derivatization peak. The reaction is very fast, occurring on the order of tens of milliseconds, and the reaction with water to eliminate the excess reagent is complete in a matter of seconds. The separation that we chose to do is based on ultra-performance LC. UPLC is a class of separation science which is based upon using very small particles of packing material on instruments that are optimized to minimize dispersion so that you take advantage of those small particles. The consequence of ultra-performance LC is that you get improved resolution and sensitivity because of the reduced dispersion, and usually an improvement in throughput. It works for all kinds of chromatographic applications, but it certainly is very suitable for the reverse phase techniques that we wish to use here. The method that we developed comes as a essentially turnkey application solution, including the instrument, the software controlling the instrument with its methods, a startup kit that includes the chemistry for doing the derivatization and for doing the separation. It can be used with the ACQUITY H-class and the ACQUITY UPLC. The method was originally developed with the UPLC and was then extended for use with the H-class and the H-class biosystems. The two methods use the same chemistry kit and follow the same protocols. The separation on the two instruments is virtually identical. There is a shift in retention of a few tenths of a minute in the later part of the chromatogram. We constrained ourselves in the development so that we would use the same chemistry on both instruments and to adjust the method so that we could get the same analytical quality. The kits used for the instrument can be used with any of the common detectors. We most often use the method with a tunable UV detector because all the amino acids have the same derivatization group. Sometimes we will incorporate a photodiode array detector or a fluorescence detector because the instrument may be used for other applications where those detectors would be desirable. We show the results here with the mixture of amino acids commonly assayed in a cell culture assay where the number of amino acids is somewhat larger. The application kit used for the two instruments is essentially the same. With the UPLC, you have a couple of hardware changes because the column heater and the injector mechanisms are different, but all of the chemistry and all of the results can be expected to be the same. The method itself is suitable for 4 general classes of applications. We use it for cell culture, which is perhaps the more challenging because of the large number of amino acids. For hydrolysates of purified proteins. We also include the alkylated cysteines that are used with purified proteins to protect the sulfur-containing amino acids. And then we also use a slight modification of the gradient table to analyze the compounds commonly found in feed samples. The complete system for these applications, whether it's an ACQUITY, UPLC, or an H-class, represents a turnkey solution for analyzing any amino acid sample. The intent is to provide the same result on any instrument in any lab on any day. The applications that we have good support and good development for include protein identification and quantitation. We monitor the nutrient levels in cell culture media, and of course we can analyze food and feed samples for their nutritional quality. So let's think a little bit about what the properties of the amino acid analysis are. Looking again at our chromatogram, we have the good separation of the amino acids where the peak identification occurs by retention time and peak quantitation occurs by the area of the peaks. The retention time reproducibility is more than sufficient for identifying the peaks. The way that I usually look at this is at the standard deviation of the retention time. The peaks are, in every case, more than 3 standard deviations apart, so that there is little or no chance of misidentifying a peak. The area reproducibility on multiple injections from a single sample is in the 0.1% RSD range, which gives us more than adequate quantitation and makes the chromatographic assay the smallest source of error within this particular assay. The linearity of the method extends to almost 4 orders of magnitude for all the amino acids. Finally, one of our desires in building this method was to make sure that it was compatible with electrospray MS. The use of MS is a specialized detection that would be used to confirm peak identity during validation and to identify peaks during a investigation of a sample that had an unexpected component. With the mobile phase that we've developed, you can do electrospray ionization and get the mass of each peak that corresponds to the mass of the derivatized amino acid. We wouldn't normally use this as a routine detection technique. There is no gain in sensitivity when we go to MS as compared to UV, and the identification of the peaks in most samples is certain from the retention time of the peak. However, I can show you an example of how we might use this and how we have successfully used the technique to solve a particular problem. We received a sample from one of our users that was a cell culture sample, and when we analyzed it, it had a perfectly good distribution of amino acids with the exception of a huge amount of serine, totally unreasonable for the cell culture sample that we had received. When we looked at this using the MS, we could see all the same peaks, but we still had the large signal for the peak at the retention time of serine. When we looked at the spectrum of that peak, however, the extracted spectrum of the large peak in the sample corresponded to the molecular weight expected for tris. We were able to confirm that the large peak in the sample was the tris buffer by derivatizing a pure standard of tris. The peak corresponding to the known tris co-eluded with the odd peak in the cell culture sample, so we needed to get a fresh sample from that media that had not already been diluted with the tris buffer. We've been looking at some of the basic properties of the method as such, so we can go on and consider how we might use it in a variety of analytical techniques. Basically, amino acid analysis is applied to two broad classes of samples. One is the components of proteins, the so-called bound amino acids, and the other is metabolic amino acids, which would be free amino acids, things that are not only building blocks, but sources of nitrogen for the protein. Considering first the bound amino acids, we're looking here at a technique where the protein or peptide sample is hydrolyzed. We would use this to confirm the identity of the protein, to determine the amount. To successfully perform this analysis, we need to unequivocally identify the peak. We need something on the order of 2 orders of magnitude linear dynamic range, which would exceed the range that we would ever observe in a real protein sample. When we hydrolyze the protein, we are having the limitations associated with the destruction of tryptophan, the conversion of asparagine and glutamine to aspartate and glutamic acid. Hydrolysis is probably the greatest source of variability in an amino acid analysis and is also the most subject to matrix interference. A commonly acceptable goal is to get a compositional accuracy on the order of about 5% for all of the amino acids within the sample. We have used as a model system bovine serum albumin so that we have a standard protein that we can compare our measured composition to the known composition from the sequence. This is the amino acid analysis of the bovine serum albumin hydrolysate. We looked at the linearity of the method over the range of 20 to 1,000 micrograms per mil, and those different levels are overlaid in this chromatogram. If we zoom in to the baseline, you can see that the smallest peaks are still perfectly symmetrical, easily integrated, and have very good signal to noise. You also start to see some of the small unidentified peaks that are often observed in the analysis of real samples and represent either side products of the hydrolysis or background from the environment. The comparison of the compositional analysis to the true value over, in this case, 75 independent analyses conducted over a period of several days were well within that 5% limit for all the amino acids observed in this hydrolysis. The use of this technique is used not only for the identification of the protein, but also for measuring the total protein concentration in the sample. If we look at a series of 25 independent determinations, we get a concentration, a total micrograms of amino acid per mil of hydrolysate, with a standard deviation on the order of 7% for these 25 independent assays. The totality of the error is primarily in the pipetting. If we incorporate an internal standard, the standard deviation comes right down closer to 1%. That's the level of reliability that we can expect in measuring the total concentration. Now, the examples we just looked at are all things that speak to how well we can determine the amino acid composition of a purified protein or a protein that is in a formulation. It is also interesting and important in the biopharmaceutical industry to be able to assay free amino acids. The free amino acids are certainly used as nutrients for the growing cells in culture. They are used by the cells not only to build proteins, but also to move nitrogen particularly, but also other functional groups through metabolic pathways. They become very good biomarkers, and they can, in fact, provide metabolic fuel, pure energy fuel for the growing cells. One of the things that is most important is to monitor a growing cell culture media. Here, we're looking at the free amino acids both as a nutritional element that limits the growth, controls the growth of the cells, but also as a measure of the waste products being produced by the cell metabolism. In this case, we're looking at a larger set of amino acids than would be monitored in a hydrolysate. First of all, we're looking at the amino acids that are damaged in acid hydrolysis but are used by proteins, things like glutamine and asparagine that are critical to the cells but would not be present in a protein hydrolysate. We're looking at the composition of batches of media as part of the optimization of the growth cycle, but we're also looking at changes over time to determine when the media needs to be refreshed, when the cells need to be fed. So we're looking at the composition, and we're looking at changes in the composition as a function of time. The additional amino acids that we monitor include things like hydroxyproline, asparagine, taurine, glutamine, and so on through the list. Some of the things are present as more or less adventitious or unexpected components coming from the media itself. Some of them are critical nutrients, and some of them will be produced by the cells as waste product. This is an example of a cell culture growing over a 6-day period. You'll notice that the amount of glutamine goes down from one day to the next, and the amount of alanine goes up from one day to the next. The changes in the composition of the media indicate the changes that must be made to optimize the growth cycle. Now, in doing these kinds of assays, we are doing a derivatization of the amino acids that is a manual derivatization. And we have investigated the possibility of automating this derivatization for the convenience of the user. The steps in the derivatization protocol always begin with either hydrolysis or deproteinization of the sample. You then add an aliquot of that sample to the reaction vial. You add a measured amount of buffer to control the pH of the derivatization. Then you add the reagent to the vial and then inject 1 microliter of sample. When this is done manually, it involves a human doing all those pipetting steps. We've used a Tecan Freedom EVO instrument to automate the derivatization process. The bed is laid out with the supply of pipette tips and then plates that contain vials with the sample, vials that contain the reagent, and vials that will contain the derivatized sample that can be then transferred to the instrument. There is a workstation for capping the vials and a workstation for shaking and heating the vials. The user places onto the instrument bed the samples, the autosampler vials, and the borate buffer and AccuTag Ultra reagent. What the instrument does is to pick up an autosampler vial and remove the cap. It adds borate buffer, then it adds and mixes a sample, and then it adds and mixes the reagent, caps the vial, and then returns the sample vial to the rack. When it's added the reagent to all the vials that are scheduled, it heats the entire batch of samples all at once. The user then takes that rack of completed samples to the ACQUITY instrument. The results, of course, of this process are the same. Just to look at some of what the steps look like, the pipetting steps occur primarily in the system in a vial holder where the cap has been removed from the vial, and the mixing occurs by pipetting the samples up and down. The robotic arm puts the cap back on the sample and transfers the sample vial to the capping station. The results of this derivatization are, of course, identical to the results that you get with a manual derivatization. What we do observe is that the Tecan is more reproducible than a skilled manual operator. This is matching sets of 6 derivatizations done by a fairly skilled operator, and the same set of samples, same proportions done with the Tecan. The results are the same, but the reproducibility is somewhat better using the Tecan. Again, almost all of the variability is in the pipetting and is reduced by the incorporation of an internal standard. The other area that has been of great interest to our users and has been of concern over the years is the preparation of the samples, particularly the hydrolysis of the samples. When amino acid analysis was first being developed many years ago, the run times were very long and relatively few samples could be run on each day. As analytical methods improve and the throughput of the analysis has become better, the limiting step has become the hydrolysis of the pure proteins to prepare the sample for the analytical method. The hydrolysis itself is basically a technique that takes 24 hours. The sample is boiled in 6 normal HCl overnight or up to 24 hours, and that process can be done either in a vapor phase or in a liquid phase. The vapor phase is much preferred for high sensitivity work and is very suitable for very pure proteins. The protein sample is dried in a sample vessel, very small test tube that can then be placed in an oven. The reaction vessel, which we can think of as a reaction bomb, might contain a dozen samples or more, and a common volume of 6 normal HCl in the bottom of the hydrolysis vial. That entire larger vessel is heated together, and the vapors of the HCl hydrolyze the amino acids. This is very good for high sensitivity because you're minimizing the liquid that you add to the sample. At the same time, it is not the best method for complex samples. If the samples are complex, the vapors of the HCl do not penetrate the solid surface of the complex samples and may not provide a sufficient excess of acid to complete the hydrolysis. In that case, we would return to the classic liquid phase hydrolysis, where the sample is suspended in a 10 or 100-fold weight excess of acid. You then seal and heat that. Seal that under a vacuum and heat it again to 105 degrees. Because you're adding the acid directly to the vial, you are more likely to add some background amino acids to the vial. The liquid phase and vapor phase methods can give essentially the same results. The standard liquid hydrolysis, one of the devices that we found very convenient to use, is an Eldex hydrolysis derivatization workstation. You would put 90 microliters of HCl, including some phenol, into the fired 6 by 50 millimeter tubes. Generally, the best way to clean glassware for use in amino acid analysis is to heat in a glassblower's oven overnight. After you put the HCl in the tube, you add the samples to each hydrolysis tube, a small number of micrograms. All the hydrolysis tubes go in the same reaction vial with a volume of liquid in the bottom of the vial. You purge the vial with nitrogen and then evacuate it and seal it under a vacuum after the third purge with nitrogen. This entire vial then is placed in the oven that's built into the back of this device for 105 degrees for 24 hours would be the most common conditions. You can use the same device for vapor phase hydrolysis. However, we've been investigating the use of microwaves as an alternative to the hydrolysis procedure. We can do microwave as a vapor phase procedure where we put the sample into the same 6 by 50 millimeter vials and dry the sample into the hydrolysis tube. We then place in the volume of the larger hydrolysis vessel, the 6 normal HCl, and turn on the microwave. In this case, we can now, instead of doing the hydrolysis overnight, we can do the hydrolysis in something on the order of 15 minutes, so that now the rate of hydrolysis is more compatible, more similar to the rate of the analytical method. We also use this device in a liquid phase where we put the acid directly into the vial, just as we did in the liquid phase hydrolysis reaction vessel. The protein concentration observed as a function of time is very much stable after 10 or 15 minutes with a 165 or 170 degrees centigrade hydrolysis. The changes in the analytical results do reflect the various time and temperature, as you would expect for any sort of chemical reaction. The proportions don't change very much. The absolute amounts do increase with increases in time and with temperature. We can get the same results doing liquid phase hydrolysis in the oven and in the microwave. We just get them 23 hours sooner. And we get the same results doing liquid and vapor phase hydrolysis for purified proteins in the microwave. We have investigated the effect of the sample formulation. In most cases, we would like to be able to measure the protein concentration of our biopharmaceutical as we formulate it, so that there's buffer, so that there's stabilizer, so there's sugar being present. All of these things have the potential to interfere with the amino acid analysis. We tested this with myoglobin and BSA so that we would have well-characterized proteins. We cleaned the vessels and did the vapor phase and liquid phase hydrolysis of the formulation. For the microwave, we used the smaller amounts of material. When we compare the liquid and vapor phase hydrolysis on the formulation, it's immediately obvious just from casual inspection that the liquid phase hydrolysis gives a higher yield of amino acids, and you can see that there is also some change in the proportions reflecting a more complete hydrolysis using the liquid phase. So the vapor phase and liquid phase for proteins in a formulation clearly shows that the liquid phase, because it distributes the samples better and provides a larger molar excess, gives you a more complete and more accurate analysis of the protein. And we can confirm this by looking at the composition. The compositions do change slightly when we go to the liquid phase, but the biggest change is in the absolute amount of the amino acids. The absolute amount comparison shows that the yield is on the order of twice as great doing liquid phase hydrolysis. So what we've been looking at is the application of our UPLC amino acid analysis solution to a variety of biopharmaceutical problems. We showed that we can get an accurate and precise measure of the amino acid composition. We showed that we can get a linear response and that we can use a electrospray mass spectrometry system to identify peaks for purposes of validating the method or for purposes of investigating an unexpected result. We did show an example of automating the derivatization for improved precision, and we also showed some of our early investigation of microwave hydrolysis to provide an alternative to conventional acid hydrolysis. I'd like to thank you all for spending this time with us today, and I hope this material was interesting and perhaps useful. I'll turn it back now to our moderator. Back to you, John.
Tom, thank you very much for that broad but very incisive overview covering the use of novel approaches to amino acid analysis for a wide variety of applications. And it looks like you've certainly struck a chord with our audience as we received a number of some really interesting questions. So why don't we take the first one? Okay, Tom. Let's take this first question here. How do the results for this method compare to some of the other techniques for amino acid analysis?
Thank you, John. The comparison to other techniques is a very large question, of course. In general, where we have been able to run the same samples by our method and by one of the alternative amino acid analysis techniques, we generally fall within 1 or 2% for all the values of all the amino acids. We've put more emphasis in our development and in our evaluation on using samples where we can know what the right answer is and therefore compare whether we're getting, how closely we approach the true answer. Our best source of that kind of experiment, of course, is a purified protein where we know the sequence and therefore we can recognize the expected amino acid composition. When we do that, we introduce all the variability not only from the amino acid analysis method, but anything in the hydrolysis and other sample handling. When we have done that on a reasonably careful basis, we typically are better than 5% agreement for all the amino acids. The amount of variability depends on the amino acids and how well you control for destruction during hydrolysis. The general perception that you would find in ABRF studies is that for a comparison to a true value, you can expect to be within 5 or 10% of the true value, and we certainly get better results than that. Hope that answers your question.
Thank you, sir. And Tom, is your method USP? If not, do we have to fully validate?
In terms of the USP description of amino acid analysis, we need to think about two parts of the USP monographs. The general chapter on amino acid analysis lists, I think there are 9 alternative methods that are acceptable for doing amino acid analysis, and this is one of those methods. So it can be used within the guidelines of the USP. However, in an individual monograph for an individual product, it may specify just one of the methods listed in the general chapter. If there is not a monograph governing the particular product that you're interested in, then the requirements for validation are much less. Switching from something specified in a monograph will certainly require more. In my experience with both the USP and the EP, people who are governed by those sort of regulations, the level of validation that is required and executed tends to be very much influenced by the individual investigator, the individual company that's making the product. So, yes, our method is a USP method, and some level of local validation will be necessary, and the extent of that tends to be in the standard operating procedures of the company. Thank you.
And Tom, how do you deal with high concentrations of salt and other sample components that could interfere with derivatization or with chromatography?
That is, again, a very broad question because of the range of things that could be occurring. For example, when we're doing finished formulations and doing amino acid analysis, the material in question may be as much as 80%, 60 to 80% protein. And with the high sensitivity of this method, most of the interferences are eliminated just by dilution of the sample to get into the sensitivity range for the amino acids. In other cases, for example, if we were doing cell culture media, the concentrations become much lower relative to the intact protein in the sample, much lower relative to the salts and other nutrients. In those cases, we are careful in dealing with the salts to first, of course, dilute it to a concentration in the linear range of the assay. Secondly, we take advantage of the amount of sensitivity that's available to us, and we take advantage of the reaction occurring in an 80% aqueous environment. In general, the biggest interference that we encounter in routine analysis is, of course, the pH of the sample. If the pH of the sample is driven low, then the derivatization does not go to completion. Interestingly, there are some amino acids that are more affected than others. So the most convenient way to ensure that the pH is at the right level is to either derivatize at two levels. Because we include buffer, the lower level material gives a better yield. Or second, to add a small amount of sodium hydroxide to the derivatization cocktail. For example, instead of using 70 microliters of borate buffer, we would use 60 microliters of borate buffer and 10 microliters of 1 molar sodium hydroxide. The sample that has the additional base should have a higher proportion, especially of aspartate and glutamate. And that would be our normal way of approaching salt. The other big interference that we will encounter is either ammonia or alkylamines, things that would react with the reagent. And once again, we can recognize that it is sample components are distorting the composition by derivatizing two different concentrations of sample and expecting a change in proportion in the lower concentration if there has been interference from the sample. Hope that answers that question.
Indeed, you did. And Tom, great question. What are the best practices for quantitative amino acid analysis when one's analyzing proteins?
When we're analyzing pure proteins, or proteins in a formulation for that matter, we have to recognize that first, we are going to be operating at the sensitive end of the scale, and secondly, that not all of the amino acids behave the same during the hydrolysis procedure. Because we are operating at the sensitive end of the scale, we run the risk of contaminating the sample with environmental constituents or components of the reagent. So if we're doing a pure protein, we generally would prefer to use vapor phase hydrolysis because you add much less material to the sample. However, as I discussed in the seminar, if the sample of protein is in a formulation where there's a great deal of non-protein material, then I would go to the liquid phase hydrolysis in order to get complete dispersal of the sample and complete acidification of the sample. The many side reactions that occur during hydrolysis mean that we have to consider the preservative approach and the kinetic, if you will, approach. Certainly, to get good values for the sulfur amino acids, we would protect them by derivatization, by alkylation of the cysteine. We would also consider the possibility of getting the best analysis by splitting the sample into multiple parts. We would split the sample first to just do a plain hydrolysis and accept that in this sample there will be some variable level of destruction of the sulfur amino acids as well as complete loss of the tryptophan. Then the other part of the sample could be subjected to performic acid oxidation. The performic acid oxidation is the best way to quantitatively convert methionine, cysteine, and cysteine into very stable amino acid, modified amino acids, if you will, so that we would analyze methionine sulfone and cysteic acid. If we also wanted to use an analysis of tryptophan, we could use a third aliquot of the sample and do the hydrolysis in 4-normal methane sulfonic acid. The third hydrolysis in the methane sulfonic acid will give us good results for tryptophan, not as good results for some of the amino acids. So we would use all three analyses to get the best composition to include the fragile amino acids. In addition, some of the amino acids are destroyed over time during hydrolysis, most specifically serine, threonine, and tyrosine are gradually destroyed. At the same time, amino acids that are in a very hydrophobic bond are released very slowly during the hydrolysis, so they increase over time. So we have some amino acids that go down with time, some amino acids that are likely to go up with time. For a very, very long time, it's been an accepted practice to do the hydrolysis for 3 time periods, and what we've generally used in the past for a HCl hydrolysis is 24, 48, and 72 hours. We can then extrapolate the threonine, serine, and tyrosine amounts to a 0 time to account for the destruction, and extrapolate usually isoleucine and valine to a stable time. We look for the amounts to stabilize to get the best analysis for that. So it's a very long answer to a very short question. Thank you, John.
Thank you, Tom. And Tom, we have time for one more. How can you recognize that the derivatization has reached quantitative completion?
In any derivatization, in any part of analytical chemistry, of course, that is the hardest thing to recognize. In the case of this amino acid analysis reaction, our best indicator of completeness of the derivatization is to take advantage of lysine. Lysine has two amino groups, so that in the normal reaction it derivatizes at both the alpha and the epsilon amino acid. If the reaction does not go to completion, the reaction will leave some monoderivatized lysine, which elutes as a separate peak between histidine and serine, and we can monitor the completeness of the derivatization reaction by the amount of the monoderivatized lysine peak. Very straightforward. Hope that helps.
Perfect. Thank you, Tom. Well, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. Hey, I want to say thank you again to Tom for his outstanding presentation, and thanks to our audience for your attention and for your very thoughtful questions about a number of topics brought up during the webinar. And thank you to Waters, whose support made this webinar possible. Bye for now.